We, as humans, have done some pretty incredible things, from the creation of language to walking on the moon. But there's still a lot of things that microbes just simply do better than us. But we're a clever species, and if they do it better, well, we'll just let them. And that's where we are now. We have already hijacked the biological machinery of some yeast and fungi to do our bidding. From cleaning up oil spills, to making biodiesel, synthesizing medicine, and even brewing beer. But now we're running into problems. For example, what if that thing you want to make is toxic to the microbe? Or oftentimes the microbe will simply stop doing what you've tasked it to, because why should it spend energy making a product for you and no benefit to itself? And so for some applications, we need a new organism. I would argue a new form of life, designed from the bottom up to do exactly what we want and programmed for nothing else. That way we don't have to walk this fine line between keeping our microbes happy and healthy and having them do what we want. I want to make a man-made cell. Now luckily I'm not alone. Synthetic biologists all over the world have chosen just one piece of what makes a cell tick and are working to remake it for use in an artificial cell. Now the piece I have chosen is DNA replication, making a copy of that genetic code. So that way when our synthetic cell divides into two, both new cells have an instruction manual for how to be a cell. Now most organisms use DNA as a template to make more DNA, and they have complex machinery needed to pull those two strands apart so they can get in there, read it, and make a copy. But something to keep in mind is if I want my technology to be versatile enough to work with other technologies, is that it needs to be simple. And so instead, I drew inspiration from some of the simplest biological entities out there, viruses. Some viruses forego using DNA altogether. They much prefer RNA. That way they don't have to worry about pulling strands apart. RNA is already single-stranded, it's already open, ready to go for a copy to be made. And so remodeling this viral machinery would be much easier than trying to hack the human or even the bacterial equivalents. And so in short, I'm working to make a technology that makes DNA copies from RNA for use in a synthetic cell. And when I do, I'll combine this technology with those of the other labs to make something far greater than the sum of its parts. And for the second time in Earth's history, to make a cell that did not come from a previous cell. Thank you.